Hello students, welcome to the course Biomolecular Spectroscopy, and in this course we are discussing the application of UV visible spectroscopy. So, till now we have discussed the basic concepts of UV visible spectroscopy. How are we going to use for biomolecules? We have seen the multiple component biomolecules, how we can deconvolute if the signal coming from more than one chromophore. So, we had looked the multiple component analysis, and this was used for determining if the signal is coming from one or multiple component. We also discussed the principle of additivity, right. So, and how we can use the statistical component approach to analyze the multiple component analysis. We have in greater detail discussed the blue shift or red shift, and we also gave some example where we can use the UV visible spectroscopy, right. So, till now this we have discussed. Now, today I am going to discuss an important topic which is not only related to the UV visible spectroscopy, but related to human life, right. How we see it, what is the science or biological science behind our like visualization, perceiveness and all those, and what the nature has given us to detect, analyze and decipher. How sophisticated a spectrophotometer we have, that is what we are going to look at. So, I am going to introduce to you the vision and photoreceptor, right. What is the molecular basis of light detection, where and how we can detect it. So, as we know, in our eyes there is a spectrophotometer, right, and this spectrophotometers enables us to see different colors, and then these colors. So, if you look at these colors, we can perceive, our brain analyze and give us response, right. So, where those are located? These are located on something called photoreceptor cells. These photoreceptor cells are in the retina, right. So, retina in the eye, there is a component called retina, and there are different kind of cells that has this. So, say rod cells, right, makes us see in the dim light, whereas the cone cells helps us for looking at the bright light or color vision, right. So, what is the composition of this photoreceptor cells? We have something called rhodopsin, it is a protein, Opsin is a protein, opsin is a class of protein called, it is called GPCR, G coupled protein receptor, right. So, you know there is an important component or important class of protein which is called GPCR. About 1/3 of our proteins are membrane protein, and in this the 7 transmembrane helix protein is called GPCR, G coupled protein receptor, and you will be surprised to know that this GPCR are the most important component of drug discovery, because lots of regulation happens through GPCR, and our opsin protein which is in question at the moment for vision is also a GPCR. So, this is a protein, and what is the chromophore that absorbs the light? It is called 11-cis retinal. That is a chromophore which is nothing but a vitamin A derivatives, and that vitamin A derivatives is a chromophore that absorbs and do something, some chemical reaction, so that we can see that or perceive that color. So, you see that is why it is very well known in the community that consuming vitamin A is very important, and a carrot which comes in the winter has lots of vitamin A. So, that means like a consuming carrot is very important for your health, and that essentially your vision is improved. So, coming back to the course, so what we have? A protein called opsin and a receptor which, or a chromophore which is just, so this is a receptor, opsin protein is a receptor, and there we have a chromophore called retinal. right. So, this linkage is covalent bond of a specific lysine residue from this opsin protein, which is lysine 296 in a transmembrane helix 7. So, as I said, there are 7 transmembrane helix. So, 7 transmembrane helix like something like this, and that sits in the membrane environment, and one of those helix 7 is proteinated by some chemical reaction called shift base, and that links it. So, function of this in dark, this 11 cis retinal keeps opsin inactive, and when it absorbs the photon, the photosynthesizer in all transform happens, and that initiates the visual signal. So, the cis trans, so you know cis means like this, trans means like this, the bulky group it rotates. So, that cis trans isomerization happens. So, if you look at here is our 11 cis retinal chromophore, that is a opsin protein, and that is a pigment rhodopsin, right. So, that is the protein that we are going to use it. So, this is called opsin shift. So, the protein tunes the light absorption, right. So, what kind of light we are going to do. So, in free 11 cis retinal it absorbs around 440 nanometer, and rhodopsin, right, this is just our chromophore, and rhodopsin when it binds to opsin protein it shifts to 500 nanometer. So, this is the shift that happens, and that is tuned by this protein. So, let us see how it happens. So, in free retinal absorption, that means this chromophore, or you can see this chromophore that we had here, this is the chromophore, right. So, the free chromophore protonated shift base of the retinal is an inert in solution and absorbs maximally at 440 nanometer, right. And within the opsin protein, this absorption maximum shifts to significantly around 500, that is a green blue light. So, the definition of opsin shift is the difference between the lambda max of the free chromophore and the bound chromophore. Free chromophore just in solution, and bound chromophore when it binds to opsin. So, the mechanism is a specific electrostatic and steric interaction modulates this energy gap between the ground state and excited state of retinal, and that is how the absorption shifts. So, if you remember basic concepts what we have seen, this is our absorption, right. So, this is ground state, this is excited state. So, in free this equivalents to 440 nanometer, and something happens upon this interaction, this shifts, and then after shifting this lowers down, right. So, in lambda max in free cysteine it is a 440, and once it bounds it becomes 500. So, here is the interaction that we are showing now of the opsin protein with the like this 11 cis retinal molecule, right. So, this is the interaction that is happening. So, what is the, like if you look at here, what is here, positive negative charge, negative positive charge, and here is a glutamate residue. So, this critical negatively charged amino acid like glutamate 113 is positioned close to positively charged shift base of the nitrogen, and that the interaction negative charge with a positive charge, this stabilization of negative charge is strongly stabilized by the ground state of the positively charged, right, PSB. Now, the effect of that happens that increase in the ground state stabilization leads to a blue shift effect compared to the neutral environment, and that increases the energy needed for excitation. So, lowers down the energy. Now, this is the stabilization that is happening here because of the glutamate interaction with this nitrogen. Now, what are the other residue that comes? So, aromatic residues are there. So, that like if you look at the lining of hydrophobic pocket, they stabilizes the excited state, right. So, the hydrophobic pocket here if you look at stabilizes the excited state via dispersion and dispersion force contributes to redshift. So, now it was just in the here stabilized by the glutamate, and in this case excited state stabilized by the like a aromatic amino acid, that leads to redshifts. So, polar residue and H bond, the presence of hydroxyl group like serine, threonine at the key position which is in the opsin protein like 180, 277 and 285, fine tunes the system through a specific hydrogen bond. So, in this receptor and the ligand that the chromophore binding happens, there are multiple forces that comes into picture. If you have done the protein structural aspects, so in the protein the hydrophobic interaction happens, the London dispersion force happens, the hydrogen bonding is there, electrostatic interaction that happens and disulfide bonds are there. In this case, these all forces helps in stabilizing ground state or excited state. So, your presence of hydroxyl group which is present in the amino acid like serine and threonine that are present at the position 180, 277 and 285 fine tunes the spectrum through a specific hydrogen bond, and the entire protein pocket acts as a sophisticated solvent where the minor change in the polarity, minor change we have seen the effect of polarity on the absorption. So, minor change in the polarity shifts the spectrum that is absorbed, right. So, that is what happens. What we have other than that, let us so color vision, how we can see the different colors. So, that is the two only wavelength I talked to you, one is 440, another is 500, that is. But in that case in our eyes there are multiple opsin genes which expresses multiple proteins. So, this multiple opsin genes that we have, human have a rhodopsin, right, rod, and 3 cones that are photoopsins. These 3 cones, rod for dim, cones for bright light, right. So, we have 3 cones. So, SWS1 that is tuned for the blue UV visible, that has a lambda max of 425. Then we have a MWS that is tuned for green light and absorbs at 533, and then LWS that is tuned for the red light that absorbs at 560. So, these are the 3 different opsin genes in the cone cells. Now, this shared chromophore, we have only one chromophore, all pigments have identical chromophore. The chromophore was what? 11 cis retinol. The spectral difference is solely due to opsin protein sequence variation, right. So, the protein sequence, although they have lot more similarity, but there is a critical variation, and that critical variation introduce the environment change in these 3 shifts. So, different in opsin genes allows our eye to cover entire range. So, look at the local environment around this chromophore actually tunes what kind of absorption they will have, and that different the protein, this 3 protein we talked about allows our eye to cover the entire visible spectrum using just one type of chromophore. One chromophore depends upon what is the local environment, can absorbs at different wavelength, and that is helpful in looking at the different colors. So, this is our absorption. If you look at SWS for blue, MSW for green, LSW for red, and this is our rhodopsin, right. So, these are the 3 different chromophore that we have, right. So, LWS and MWS tuning mechanism that happens by something called 5 site rules. The spectral tuning happens with this. So, if you look at these 3 protein, these 2 protein, let us see LWS and MWS. For red and green, they pigment, this pigment shares about 96% identical, and they absorbs like this 96%. So, 4% are basically critical. Those 4% differences makes this retinol to absorbs around 30 nanometer apart, right. What are the key sites that makes this differences? It is accounted only by amino acid difference at 5 specific site, and those sites in the protein are 116, 180, 230, 277 and 285. These are these. So, what are there? The tyrosine, if you remember the structure, it is a aromatic ring and OH group. Alanine, it is a hydrophobic amino acid. Phenylalanine, it is a ring, right, and alanine again hydrophobic, and again 285 is alanine. So, here are those residues, right. So, MSW binding pocket with 11 cis retinol is like this, and LSW cis is like this. So, these are the critical amino acids this is for red, this is for green. So, look at most of the amino acids are hydrophobic, only L116 is having OH group. So, only the subtle differences in the background of this retinol is making them absorbs about 30 nanometer, right. So, SWS spectral tuning happens like UV visible versus violet sensitivity. So, this is the structure, phenylalanine 86 is here, and opsin single amino substitute basically shifts it. So, if you look at SWS class, it is a shorter wavelength sensitive opsin, typically absorbs in the UV 355 or violet 410, that is the range. Single point mutation in this, where is the mutation happening? This phenylalanine is changing to valine and responsible for large spectral shift, right, between here to here about 65 nanometer. The mechanism, what is happening, if you look at this is phenylalanine, bulky amino acid, there is a valine, smaller amino acid. So, this single mutation, what it is doing here now, up if you look at little bit of his structure, 11-cis retinol is surrounded by phenylalanine, like a it has aromatic cloud, here is a hydrophobic cloud. So, that is the change in the environment is governed. So, if you look at here, slightly positive on the surface, slightly negative on the center of an aromatic, and this is completely hydrophobic. So, this single point mutation can determine single point mutation from here, right. So, from here 355 to 410 shifts can happen. So, it determines that whether animal can see in UV visible light or not, highlighting the power of precise change in the protein sequence. So, you see the beauty, right. So, nature how beautifully tunes it. Just by changing one amino acid, it can change what kind of light an animal is going to absorb. So, just to give you an example how evolutionary adaptation happens, right. So, convergence of evolution with spectral sensitivity. It also determines the habitat. So, deep sea ocean, what is the color of deep sea ocean? It is a blue. So, blue shifted sensitivity you require, right. So, about 475, this fish has that, right. And now terrestrial opsin, standard sensitivity, birds can see that, right. So, birds can see that, it is about 500 to 560, and freshwater opsin, red shifted sensitivity, right, 420 to 540, these fish can see it. So, you see depending upon what is the need, nature tunes that with the same protein just by little bit changing in the amino acid. So, species tune their opsin to match the ambient light for this deep sea opsin for the fish living in the deep sea, right, or freshwater it is a red. So, that is what tuning is happening here. So, convergent evolution, spectral similar spectral shift in the distantly related species often involves the analogous change in the same general region of the protein, right. So, one amino acid change can do that, and mechanism, this natural selection favors the opsin variants, and the maximum photon capture in a specific environment can happen because of this. So, in nutshell, evolution has optimized the opsin, same protein changing only few amino acids or maybe one or two amino acids, tunes the animal to see in that natural habitat. So, evolution has optimized the opsin for specific environmental from deep sea to the forest canopy, right. Now, coming back to the A1 versus A2 chromophore system. So, what is the alternate chromophore, A1 or A2? What are those? Basically, if you look at A1 retinal, it is 11 cis retinal, A2 retinal is 3,4 didehydroretinal, and that is the structural change that happens. So, this is like a extended conjugation system basically shifts it. So, let us go back. So, vertebrate use two form of vitamin A derivatives, chrome 4 which is coming from this whatever we are discussing 11 wall retinol, versus the 2A2 which is 11 cis 3,4 dihydroretinol, that is the A2. So, here is that difference, this is also, this is the structure, right, cis trans, you can, this can be also done. So, A2 effect, so A2 what it is doing, A2 has an extra double bond, right, if you look at here, conjugated system and significantly rate shift for absorption of maxima by 20 to 40. Now, this can do dynamic tuning. So, many aquatic animal dynamically tunes A1 or A2 ratio to match the rate shift in the turbid or deep water. So, nature again has provided the animals the tunability by just switching between these receptors. So, some animal use second mechanism, switching their vitamin A type A1 or A2 to tune their vision to a specific aquatic light environment. That fine tuning not only done by the protein and changing the environment, but also by little bit tuning of the chromophore. This larger the pi conjugation system helps in fine tuning the chromophore. So, that is what it has. A1 system high sensitivity, low noise, it is a photosensitivity, low thermal noise, good for dim light vision. Now, red shifted absorption, the higher thermal noise, compromise for red light shift environment, this is in red, this is in blue. So, evolutionary compromise between a spectral absorption and detection preferences can be done by A1 and A2 system. So, A2 pigment has a red shift lambda max and a broader absorption bandwidth than A1 form. The sensitivity and noise, so if you look at A2 based pigment less photosensitive, exhibit more thermal noise, right. So, spontaneously it gets deactivated in the dark, and trade off. What is the trade off when we want to use it? There is a evolutionary trade off between maximizing the light capture in the red shift environment and maintaining low thermal noise for the dim. So, here is a low thermal noise, here is a high thermal noise, sensitivity is more. So, while A2 helps looking at the red light, it comes with a cost of noisier in the dark. This is quite noisy, this is sharper, right. So, now how we can model it? So, this is all from nature, and human has to learn a lot from the nature. We can basically understand all these switching from some of the modeling. So, quantum mechanical or molecular mechanical modeling can be done, or a hybrid quantum mechanical molecular mechanical simulation can be used to precisely understand the spectral shift that is happening, right. So, we can just recapitulate what nature is doing, and chromophore which is QM region, right, a quantum mechanical region, the chromophore or immediate interaction residue, like that is treated with a high level of quantum mechanics precisely. Molecular mechanism, rest of the large opsin system we can understand it, and purpose of this study that we can do the simulation to predict lambda max value and identify what specific interactions are happening here. So, we can now use learning from the nature using computational biology to understand how the switching happens and fine tuning happen, or what is the science behind it. So, computational biology can use powerful quantum mechanical molecular mechanical system to predict how a specific mutation can change the color. So, what we observe from the nature, we can basically understand using computational tools. Okay. So, now coming back to the photo transduction cascade, how we amplify it. First, I discussed that there is a chromophore which is retinol, and it is bound to a protein which is called opsin. Now, this retinol bound to a protein, but how the brain perceive it, how we perceive it, right. So, you see this is our, like a brain is our supercomputer, right, that has to get the data, process it and give you in some digestible form. So, that visual signal from photon capturing is happening by this protein. How that capture happens and then get amplified, and we understand it. So, photoactivation, all transretinal intermediate, that we can call it meta 2, triggers the large conformational change. Light got like a sign on here, that is the opsin and rhodopsin here, rhodopsin. then it has to get amplified. So, this is, as I said, it is a GPCR, G protein coupled receptor. So, now GPCR in that means the GTP comes into picture and that essentially activates it. So, it activates basically GDP to GTP, and then here meta 2 gets activated by 1 photon input, and then meta 2 binds and act as an enzyme for transduction of G protein. Now, that basically helps in signal amplification. So, once activated, rhodopsin can activate hundreds of the transducing molecule, right, in this cycle, and then chain reaction starts, and this creates a powerful domino effect where initial light signal is typically amplified at a molecular level. So, light which is shining here during the chemical reaction, that chemical reaction is getting amplified, which is called domino effect, and that basically this molecular level change is creating a signal which is captured and then amplified for us to visualize it, right. So, that is what happens, the visual signal, it is a happens and neural transmission happens. So, our was a active transducin, here GTP comes and there is cyclic AMP and all those happens. So, chemical reactions, and that gets into basically neurons, right. So, that is what happens. Activated transduction subunit move to activated enzyme called photodiesterase PDE, and that channel regulation happens. This PDE hydrolyzes the cyclic AMP here to GMP and dramatically lowers the cyclic AMP level. So, all the chemical reaction happening, and that gives the signal to neurons, right. So, reduction in cyclic GMP causes cyclic GMP gated ion channel to close, and that results in hyperpolarization of the photoreceptor in the cell membrane, which sends the electrical signal to the brain for processing. So, see what is happening, light is shining. So, light doing all sorts of chemical reaction, right. So, GTP, DGP, cyclic AMP, that gives some electrical current, and that electrical signal goes to brain for processing. So, from light to chemical reaction to electrical signal, and that is how signal goes to brain, which process and see the light or perceive the light and we analyze. So, the most sophisticated spectrophotometer that we started at the beginning of class is here. Light is shining to a detector, doing lots of chemical reaction, creating the electrical signal, passing through an electrical multiplier tube or whatever you call it, neuron, and that passes to the brain for further processing. Great. So, let us stop it here with the understanding of how we can see anything and how the signal is introduced or transduced, and that is amplified and sent it to the brain for processing. Looking forward to have you in the next class. Thank you very much.